Good morning, my outstanding friends. I have heard your pleas. I have heard your cries for help. Yes, I've heard them. You want to know about opals? I will tell you about opals. Well, what are opals? You see all these colors? That's what you always think of when you see opals. You say, "Ooh, look at all those fancy colors." Well, not all opals have all these fancy colors. Let's go a little deeper into this. All right, when you think of opals, you think of six spectacular looking colors like this. This is a Nautilus, all right? That's like a sea creature. And it's a giant Nautilus, first of all. And secondly, you can see all these different colors. And you see the different green on the outside and a different color outside of that. And then it moves more to the red on the inside. Well, why is that? Why do these colors change in different areas of life? Well, let's look at a opalized heart, and it will become perfectly clear to you exactly what's going on. It's a very simple process, really, and it consists of those transition metal colors that are in water, aqueous solutions, that invade tissues. I will show you this. It's very, very simple, and they're very exciting opals because of the variety that you see. But I want to show you exactly what happens down in the what I would call the seed level. All right. Okay, we're going to go through this. It's going to take a couple of minutes, and this will give you a really serious idea of the chemistry, because this is chemistry. Oh, by the way, everybody knows my goose. I've given him a name. I call him now Caesar. Augustus Caesar. All right, now, here's what we got. What is all this colors? What's all this 3 plus, 2 plus, 5 plus? What's going on there? Well, these are what they call transition metals ion colors. Ions means that they're not perfectly neutral. They have a charge. And the charges are these different numbers. And what they do is, these are the transition metals. Right? They have deep blank holes inside of them where no other metals do, no other elements do. They fill everything up, and then the orbitals, and then they make another orbital, and then another orbital. Well, these guys fill up partial orbitals inside. So you can fill them up with these other particles, and then they change colors because light bounces back from everything. Just think about this. People don't understand light. I'm serious. No clue about light. Why is this purple, and that's green, and that's blue, and that's yellow? Why are these different colors that we're seeing? Well, they're being emitted in different frequencies here. But normally when you illuminate something, instead of this is self-illuminated, sending out these colors, normally you put something like this up in the light, and I can see all these different colors now. You see them. You see yellow, you see green, you see blue, and all those. The same light is hitting this. Why isn't it all the same color? Same light's hitting it. If it's bouncing back at us, why isn't it bouncing back? If it's all the same light hitting it, why shouldn't we get all the same color back? Well, the reason is chemistry. And what is blue here has a different little bit of transition metal in it. It has a different ability to absorb and push back. Basically, that's what everything is in colors. Clearness is a different situation where you don't have any reflectivity, you don't have any absorption. It sort of basically just goes right through. And we're going to be talking about all of this stuff, but reflectivity is what this stuff is all about. And that's why you see these different colors when you illuminate something. But let's just go through the transition metals. I'm probably making this confusing, where it's not. It shouldn't be confusing. It's just the same light is hitting everything, so we're bouncing back from different elements, and that's how I can tell what's in something. When I see red, I know it's blood. When I see black, I normally know it's primarily the other type of blood. The red blood is your artery blood. The black blood is your vein blood. And it's just because of the different states of oxygenation. That's all these are, is different oxygen states. Now, let's just read about this, and I'll try to make this as understandable as possible. I want you people to get educated. I don't want to just spout about, oh, look at these fancy looking colors and all that. That's not what this is about. This is about learning so that you can speak about these things with intelligence and debate them. Because what I'm saying might not be right. I might be wrong, and then somebody can correct me. That's fine. I have no problem with that. Now, what's going on with transition metals? These are the things that are in blood. All right? That is what's in your blood. Transition metals keep you alive. And when I say that, they are the reason that you can deliver oxygen to your lungs and pick up carbon dioxide and deliver that out. All the things in your body, urine and your different mucus and all these things are produced by bacteria that create different transition metal complexes that do the job in your body of keeping you alive. Every single molecule in your body right now has to be worked on by transition metals every second of the day, because you need more oxygen and more oxygen and more oxygen for every cell in your body. So your iron is going down to deliver that oxygen and pick up the carbon dioxide. Other things are picking up wastes out of cells. After they do their job, they create byproducts that are wastes. They got to get rid of them. Other bacteria is creating transition metal complexes that go out and harvest food from what's coming down your digestive system. If you don't have the bacteria to create the transition metals, and the transition metals to go out and do the job of transitioning molecules back and forth in your body, you're going to die, or you're going to be sick. And that's what people are sick right now, because they don't have correct probiotics in their body, breaking down the chemistry using these transition metals. That's what the probiotics do, is they make transition metal complexes that are so elegantly designed that they can go out and with a couple of little electrons they can either destroy something or create mucus or break down carrots into some edible molecules that your body can absorb. This is what is life. This is life. This is blood. All right, so that's my speech for today. Now, let me just stop for 1 second here and gather my thoughts and get ready to try to make you understand how transition metals grab a hold of molecules. They're little pinchers. That's all they do. They're just there for the ride. And they say that. They say these huge molecules are just there for the ride. Well, that's true. But what's riding on them? The things that are riding on them are the byproducts of life. They're the sustenance of life, and they're the garbage from life. You have to bring it in and you have to get rid of it, just like in your house. All right, I'm just going to get you familiar with how opals attach and what kind of tissues they attach to. This is just obviously a heart that's been dissected and cut across. Now, this is also a heart. And that was basically the same thing, only this is what they call water opal. And if I look at this in exactly the right ways, I can catch little flashes, just tiny little flakes of transition metals that are still in here. But you can see you can see the similarity of the vascular nature of it and all the tissues and so forth. Now, let's see what an opalized heart looks like. Don't forget, these are different types of tissues. You see them? So different tissues have different chemical characteristics. All right, you just saw the regular human heart. This is, again, that's the one I have here. It's been cut, obviously, and polished a bit. But you can see that was a chamber. That was a chamber of the heart, or somewhere in here. And all these are tissues and valves and all this kind of stuff. And if you look at it sideways, you can look down into it. It's almost like it's coated with glass, and you can see down into these chambers. But this is a water opal. This is a Yoa opal. Now, Yoa is a region in Australia, where they have spectacular opals. And the reason is because there was a ton of blood in that area, and it was a water opal area, but it was loaded with these transition metals that I'm talking about. Now, where did they attach? Well, that's one of the ventricles of the heart, or whatever they call it. I'm not going to try to pretend I'm a doctor just giving you all the details here, but these little things, I call them heartstrings, which I think that's what they call them. And then you have the ventricle walls, which are the constrictors that force the blood to pump through, and they pump and they suck and they pump and they suck and they pump and they suck, and that's what brings it going through your body. And one side is artery and one side is vein. Now, what happened when a guy died? Everything washed out of his body, basically, into a water opal situation. Basically, I think, quite likely, it was turned into one of these. And then blood from somewhere came into this in a watery condition. And it was still, at this point, when it was like in a water opal condition, it would still be soft and be able to still move particles around. And apparently, because it has to. So at one point or another, these molecules that were transition metals found the correct tissues underneath to attach to. Now, this area here, it looks like it was invaded and came in from this way, and all of the original stuff went here, and then it sort of blockaded itself and finished up by attaching these molecules the best it could to find the best substitutions that it could find. This is called nucleophilic substitution. So the molecules coming in want to replace, they want to invade and replace. I call it nucleophilic substitution, nucleophilic invasion. It's replacement and substitution of what was originally here by something that wants to attach to just little bits and pieces that were there. That's why they have this color, because copper or vanadium or something wants to be attached to this specific type of tissue. And every tissue in your body is different. Every single tissue. Your lung tissues are different than your heart tissues, are different than your kidneys, are different than your liver, or different than your testicles. Every single one of them has a specific chemistry, and it has a specific need for enzymes and catalysts to work with that chemistry to keep it alive, to keep the membranes flexible and supported with mucus and serosa and the catalysts and enzymes. So you need bacteria throughout your body to do the job of creating the mucus and creating the barriers that separate all these different things from each other. And when you die, that stops. The things aren't moving around anymore, and you die. So normally you would rot, but in the salty waters, or in certain conditions, let's just go with that. It could be salty, it could be acids, it could be whatever it is. It will invade the tissues, and then if something can attach to the tissues that have been invaded and stabilize them, then you end up with this, and you end up with like my goose, August. This is my Caesar. There's his feathers. Everything attaches to everything if it's in the right condition, and then it will become like this. Is they would call that basalt, probably. There's all kinds of metamorphic. They got all kinds of names for it, but all they are is names. It's 100% changing the chemistry, which was originally organic, and changing it into a stone. And what they want to find is bones. You don't find bones in mud fossils. You find complete organs, and you find everything. Bones didn't do well. So bones do change. But what they're doing now, they're chipping away at everything. They chip my whole goose's head off of here and get to the bone and say, oop, there's the bone. We figured it out. Well, they chipped off the guy's head to get to the bone. I don't think that's figuring much out. Because that's what I'm seeing now. They're talking about concretions. Everything's a concretion. Well, a concretion means a bunch of stuff just glued onto something. Well, it's flesh glued onto the bone. That's the concretion. There's tendons and flesh and everything. I've shown this very, very, I think, extremely well in my humblest opinions. So if somebody wants to confront it, just confront it. Don't just, they're running away and hiding because it's all geology to change everything. They, to them, this is just a rock that turned red and green. Now, anything biological can turn into an opal. Sedimentary stone, which is eroded stuff, they never turn into opals. The only thing is, see the squares around here? I believe this is a tree or some form of cellulose. And they have cell walls as well as cell membranes. We only have cell membranes, which is the little membrane around each cell. Well, each cell here is turned into a color, and the reason it has is because it was what's called seeds, and there'll be a crystal left in here or invaded in here, and once one gets in here and starts to set up shop, it attracts more of its like through that membrane into blue and yellow and orange and green and all these different colors, and that's how they grow. But remember this, it's only biological stuff that turns into opal. And because I can see these cell walls, I would have to say this is not a creature. This is a plant of some sort. All right, now this is opal, but in plants I usually call it agate. But you can see they're in chunks. That's because they have cellular construction, almost like bamboo. And you don't notice it in trees, but they really do have that kind of a structure. Now, this is the same sort of opalization. And you see this red little stuff here? This was all blood. This all was blood in this area, and that's why that turned that way. You won't get this until you have transition metals, and you won't get transition metals unless you have a lot of blood. See, there's a tree that got invaded by transition metals. Trees did not do well during the saltwater floods. You see how these are all expanded and broken apart, the tree rings? That's what happens when they get invaded by salt waters, and then they crystallize and they destroy the cell walls and so forth. And that's what makes these break down into nothingness. Well, this one had apparently, again, I believe they got invaded by salt waters and so forth. Some kind of chemistry invaded it and basically destroyed what was originally there, and all that was left was some of the fibers, and then it got refilled by something with a lot of blood. And that's the case here. And this one got refilled pretty quick before the fibers changed a ton, it looks like to me. I have a tree here, and this is a regular tree, same thing, but different, they're all different. And it depends on where it was and what kind of waters it was in, and, there's a lot of research to do here. This is going to be fun for a lot of people, because now you got some way of looking at these things with more of a critical eye and say, ooh, I wonder how that happened. Ooh, look at that. What's its color? How come you got blue and then you got green right next to each other? Now, and that brings up a good point. I was looking at the colors. Wait a second. All right, this is a good time to come back to this anyway. You see this iron here, Fe, ferrous, and ferrous means iron, and these are the oxide states, 2 and 3. Well, guess what? In mud fossils, the +3 state is Fe2O3. That means O3, oxygen state 3, is red. That's your red blood that's in your body. Why are they showing it yellow? I don't understand this. But I can't get down to the depth to see what these colors are. I don't have any spectroscopes or any of that stuff. And the 2 state is always black in the mud fossil, so I don't know. But these are all the different colors they're attributing to all these different oxygenation states in these different, like this is a +2 state, this is a +2 state, this is a +2 state, +2 state. They're all different colors, but they're different sizes of molecules. Again, a lot to understand about the chemistry. I'm pretty deep into that, but there's still a lot to learn in everything. But this is the key to these transition metals. The colors can vary depending on the charge on the metal ion and the number and type of groups of atoms called ligands. This is the key right here. These are the pinchers. They attach to the metal ion in aqueous solutions, watery solutions. The ions, which are these oxygenated states, form complexes, big molecules, with the colors shown to the right. That's the colors, their state. I don't know about that, but anyway. Electrons are arranged around the nucleus of the metal atom. So your metal atom is in the center, and then they have all of these, what they call ions. They're different states that can attract and give up electrons, so they can attach and carry things around with them, basically is what it is. Now, the electrons are arranged around the nucleus of the metal atom in orbitals. Transition metals, unlike other metals, have a partially filled d orbital, that's a lower orbital, which can hold up to 10 electrons when ligands are present. Some d orbitals become higher in energy than before. All right, so they have a higher energy state than their normal energy state. And some become lower, so they can go both ways. It's transition. Electrons can then move between these higher and lower orbitals by absorbing a photon of light. Well, in your body, it's basically the same thing. Light is nothing more than electrons, back to back. Light is 2 electrons, one this way and one that way, stuck together. This absorption of light affects the perceived color of the compound or complex. The wavelength of the light absorbed is affected by the size of the energy gap between the d orbitals, which is in turn affected by the type of ligand and the charge on the metal ion. So what it is a smaller molecule, a smaller atom, will have all these different states, and it can attach to all kinds of different molecules. If it's a little bit bigger, it still has these same states, but they're in a different frequency, so it can attach to more stuff. So even though you see same looking colors and so forth, they're attaching to so many different molecules. That's why they're extremely complexes, very complex. And when they talk about them, they say, don't get scared about the size of these molecules. Most of the molecule is just there for the ride. No, it is the driver. All right, most of the molecule, when you see in metal complexes, most of that molecule is the driver that carries around the waste and delivers the good stuff. That's how it works. They say it's just there for the ride. No, it's just the other way around. The other stuff is on there for the ride to go and come back to the lungs, to the liver, to the kidneys, to the wherever things have to go. And they have to go continuously, 100% of the day. 100%, 100%, 100%. You're moving oxygen, you're moving glucose, you're removing carbon dioxide, you're moving your urine and acids and salts. All of that stuff has to be dealt with. So this is a lot more than just looking at pretty looking rocks, okay? Get something out of this, if you would, please. Right? This is what I'm hoping for. People will start to engage in understanding what they're looking at, not just looking at them for, because they're shiny or that type of thing. All right, we saw the opals, and we saw that they're always going to be in cellular compartments, and this is these are cell walls. These are cells with membranes, and that's what creatures have. Plants have the cell walls. So now we've established that. Now I'm just going to run through this stuff. Here's a clam. Now everything, you see all the little sparkling things, that's all those different transition metals. Now everything that is opalized was alive at one time. This is an eyeball. You see that? You can even see the strap that runs down. This is what pulls your eyeball to move it around. And that's the different chemistry. Like I said, every bit of your body is separated by membranes and muscles and tendons and connective tissues and organs and all that stuff. Absolutely, totally different chemistry. So it's going to find something to stabilize with that's totally different from what the eyeball did, and what the center of the eyeball did, and what the muscle did, and what the tendon does. It's all different. This was interesting. Now, look at this. This is a tooth. But there's an opalized insect inside the tooth. How the hell did he get in there? How could an insect get inside a tooth and get opalized? This one threw me for a loop for a while. But if you look down the end here, that was a hole in here. And apparently, this is one of those water opals. Okay? And the water came in here and evacuated whatever was inside that tooth. Then the insect got in here. And the insect is what created the colors of the opal. It would have been a water opal, like the one I showed you, like this one here. Only he crawled up through the stem, and his transition metals caused these colors. That's what I'm taking from this one. That one threw me for a loop. We saw the opal heart, and you see all these different colors. Now, you can see that the metals are heavier than the plasma. You see the red up here? This heart was, this obviously cut in half. This heart was like this. This is the downside, and the metals sort of fell down, and the wet, not heavier metals drifted up to the top. Anyway, you can see all the different colors. It's pretty obvious. And these are, the opals only form in certain places, and they only form there because the metals are available there. And sometimes the chemistry is such that only green opals are formed, only blue opals are formed. It's the transition metals that invade. Now, look at that. I believe this was from a lung. And these are all the different colors of the lung. I have lungs here. Here, let me show you one here. That's a lung right there. Now, will you see that? I don't know. Let me see if I can get some color on this. You see all these different crystals in here. You see and actually you can see all these different the holes in there like there. That's where the blood ran through the alveoli, and there was a ton of blood in there. Blood has the crystals. Blood has the transition metals, and that's exactly what you got there. And I'm sure that was a lung. So when you see these different colors like that, even though you don't see anything on the outside, it looks a ton like it's a lung. There it is. I'm 100% sure that was a lung, and I am as well there too. You don't get all those different colors in one spot unless it's loaded with blood. Not, I mean, it could be a heart, but I say a lung. What do we got here? All right, this is the way they bead up in certain spots. It's very... Okay, we saw the Nautilus. You see that, and that's why they separate different colors. The outside is an old growth, maybe. It's more calcified. I don't know. Again, there's a lot to think about. Why is it so dark here? No. All right, this is a fingerprint or a toe. I think it's a toe, like this, or a fingerprint like this. And you can see this in the right conditions, this would have turned into an opal, just like that. And this is a toe. You see that? That's the callus on the guy's toe. I'm not kidding you. That's the callus on the guy's toe. This was the finger or the toe nail top. And this is all blood. I got the microscope going. Maybe we'll look at some of this in a microscope. But that was this is a big toe. And this is where it's attached to the guy's foot, right there. And this is all blood from underneath the fingernail. And the fingernail has turned, it's transitioned. And this is all grip skin under here. And this is transition. And that is the callus, just like you have on your toe. I don't know if it's that bad on your toe. Mine's pretty bad. And this is the wrap. Now let me show you something. This is the wrap on all bones. And that was, and all bones and all tissue. You see this right here? It's got the same exact wrap right there. You see this? You see it? I don't know if you can see that as well as I can. But that is the way they come in with angles just like that and wrap around And that gives you the ability, and they're all over the place. It's not just this one here. I hope you can see that. I never know what you can see until I get done, and by the time I'm done. So I hope you see it. If you don't, I don't know what to say. But I've always said they wrap around like a crescent roll. You ever see the crescent rolls? Well, my wife used to make them, and they wrap right around like a little triangle, snaps right in there, and that's it right there. And this, I believe, is the actual toenail, toe prints. Prints like fingerprints, that type of thing. You see there's some more here. I have mine have fingerprints in them. I have giants with fingerprints. Still perfect, absolutely perfect. Anyway, that's the toe. I believe, that's my guess on that. This was, we saw that, the tree. We saw those trees, and that's it. Okay, this is the Mitchell-Hedges skull, and you can look it up. It's disputed whether it's real or not. Well, I can tell you one thing. I'm absolutely certain it's real. And the reason I can tell you that is this was a water crystal, a water opal. Same thing as like this. However, you can still see the nerve fibers and so forth in here, and you can see that it was laying on its face, and the lip was twisted up and the nose is pushed back. But I can see every single detail, plus the jaw comes right off of here. This is all completely anatomically, exactly, flawlessly perfect. And the inconformities here in the front, which is me, it was laying on its face, down. The brain moved forward. That's why this cavity is open. You see back here, the brain would be fluffed back, you know, normally. But being on its face, it sort of drifted down. Now, what are these fibers? Well, those fibers are these fibers. That's what they are. There's virtually no question. Now, you see them all running exactly the same places, exactly identical. And this is exactly identical to the structure of a human skull, anatomically. So, and I'm sure if you looked at this correctly, you would see some transition metals flashing in the correct light, because these are different. You see this? You see the different colors here? Well, they're different colors because they're different types of fibers. Different types of fibers have different chemistry, just as I told you before. You see this green going into the blue and the orange and the red and all these different colors? They service different types of nerves. Different types of nerves have different types of sheathing around them. The different types of sheathing is what we're seeing here. And these are all running from what they call a vagus nerve, and they control different functions in your body. They're starting to understand this stuff pretty good, but I mean, who could miss this? How could you possibly miss this? That's what this is. It's water opal that never filled in with any transition metals. Now, if it had filled back in with transition metals after it turned into water opal and was all washed out of organic matter, and then blood came back in and fused this, can you imagine? It would look just like that. Very similar. But this is what you see. Look for the anatomical problems. Why is this thing pushed back? Why is his lip up like this? You have to be able to look at things with a critical eye without just a complete dismissal. And when you talk about complete dismissal, this destroyed our research. Scott Walters is the one responsible, because he looked at this on TV, national TV for History Channel, says, nope, that's absolutely fake or carved. And this is just an iron stain, which obviously iron is blood. Well, nobody pushes the flesh off of the cartilage of a nose to carve it. And now this can all be proven. There's no question. This was 6-7 years ago, and I confronted Scott. We went back and forth, and finally he just said, forget it, I'm the expert. It was 20 seconds from the first time he saw this. The first instant he laid eyes on this, on TV, in 20 seconds, nope, I'm certain it's not real. Because that's what it was brought for, to show him that it was real. And then we had a CAT scan, and we had all kinds of things. And, you know, there's no question it's real. And that really destroyed us. But anyway, that's water over Z Dam. All right, this is a giant hand, and this has been DNA tested. It's human. There's no question whatsoever. I have a whole batch of stuff from this, knuckles and fingers and all kinds of things. It's a left human hand. That's the tendon that runs down. This is the pad that runs around, the same as you have on your hand. And this is the grip skin peeling off. This is 3 feet wide, maybe a little more than that. And this is just one of them. And this is a small guy. This is about 50 feet or so tall. I have a bigger one, and it's even in better condition than this. But remember how this is peeling off the silver skin. That's the grip skin. And it's a very dense, thick, scrappy, tough skin. Now on this, it's only maybe, oh, I don't know, a 1/16 of an inch thick, or a 1/4 of an eighth of an inch, something like that. The other one is about this thick because the other one is much, much bigger. All right, this is the big finger tip, much bigger than the hand that I just showed you. This one is almost as wide as that hand was as the fingertip itself. This thing's gigantic, and that's a fingernail. Just take a second look at it. This is the bumper pad that bumps up against your next bone so they can rock against it. And these are the little blood supplies that are coming in. All right, now let's see the fingerprints. Well, here's the fingerprints. Now, this is what I call grip skin. I can't remember what the technical, but it's the grip skin. It's on your fingernails. It's on your, on your fingerprints. It's on your toes and so forth, your pads of your feet. And it's a really tough, scrappy, heavy-duty keratinized cells. Very heavy duty, and it peels right off just like this. This peeled right off. Well, that big fingertip I just showed you, I smacked that with like a little sledgehammer, and this popped right off, and it's about this thick. Well, I have it here. I could probably show you, but you don't have to see it. These are the sweat pores. Bip, 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 bip. And that's one finger ridge. That's a ridge of a fingerprint. Just like on yours, you got them all over your fingerprints. My thumb is about the width of that fingerprint. One ridge. So that was and this was DNA certified as a human, and they said it was excellent quality DNA. Because what I do is I go up inside and get blood right out of the stone, and there's blood in all of these stones. So don't tell people that you can't get blood out of a stone. They're saturated with it if you know how to get it out of there. And you have to sometimes go in to get it out. Sometimes it just comes out at you. All right, I'm pretty sure I showed you this. This is a bone. All right, and what they do is they have what they call bone foramen in them, where they have holes and blood transfers between the bone and the arteries and so forth. And they have your arteries run in, your veins run out, and you have nerves that run through, and that's how they connect from bone to bone to bone. Now, this is one of those, a bone foramen. All right? And this is a blood clot. And you say, oh, no, that's just iron oxide. Well, guess what? Iron oxide does not create fibrin, which is the scab, the backing. This is called clotting factor. That's a scab. And it creates a web that the blood vessel or the blood cells collect on so you can make sticky scabs. That's exactly what it is. So I don't think just iron oxide gives you a good explanation. And then to be able to find the actual holes that iron oxide came out of and created the fibrin, and then the blood clot formed. That's the artery, that's the vein. All right, this is, you have to really do a little more research than just looking at, oh, it's that, 20 seconds, Scott Walter. That was not good, my friend. You should make things right, buddy. My point is that you got to look at things for more than 20 seconds and examine them critically. We could find every single anatomical feature in this. There's no question it was alive at one time. And to try to explain away some of these things it's impossible. And to just be able to turn your back and walk away is not really the best way to handle something. If there's a possibility that you could be wrong, I think it should be addressed and make things right. I would.